So good afternoon, ladies and gentlemen. Thank you for tuning in to another Risk Institute online session. Today, we're especially fortunate to have with us one of our speakers, Professor Herek Clack. He's going to talk to us about preventing airborne infectious diseases, transmission, challenges and technical advances. He's joined us today from the University of Michigan. So I'm just going to give you a brief introduction of Professor Herek Clack. Herek Clack is an associate professor at the Civil and Environmental Engineering at University of Michigan. At his university, his group focuses on chemical and biological aerosols and the interactions of electric fields and electrical discharges. He's also the co-founder and CEO of the startup Taza Aya LLC, a company doing incredible work specializing in non-thermal plasmas to kill pathogens in the air. Their current innovation, the direct air treatment module, treats air to achieve safety of social distancing without masks. He served on numerous National Research Council committees addressing environmental issues ranging from the implications of changes, the regulations governing power for plant emissions, to the safe and ethical thermal destruction of both conventional munitions and the chemical warfare against agents by the US military. He's the recipient of many awards, including but not limited to the 16th Distinguished Young Alumni Award at MIT, the NSF Career Award, and the Kenneth M. Reese Outstanding Research Scientist Award at the University of Michigan. He's also the Vice President and a member of the Board of Directors of the International Society for Electrostatic Precipitation, serves in the Mitigation and Control Technology Working Group, Awards Committee, and the Representation and Equity Affairs Committee of the American Association of Aerosols Research. He started his journey in academia with a bachelor's degree in aeronautical and aeronautical and astronautical engineering from MIT, and a master's and PhD in mechanical engineering from the University of California, Berkeley. So without further ado, Professor Herek Clack, would you like to share your slides and begin, please?
Okay, thank you, Francis. Appreciate that voluminous introduction. Really, feel free to interrupt me or ask questions along the way. We have an intimate group here, so I'm happy to have a conversation more than didactics. So as you are aware, this is the subject of my talk, basically. And in the images I have there, I'm trying to convey, the many varied applications of the type of work that we do in our lab. And so, pre-2019, myself and my colleagues and my grad students, have been working on airborne infectious agents, but in order to properly position what we do for audiences, we often would have to give background on why they should care about airborne pathogens. We had to, you can see here, we had to make a comparison to other highly impactful diseases with high mortality rates. We had to remind people of, historic pandemics in the past and that they possibly could reemerge in the future. And I have a picture here. This is probably not familiar to you in the UK, but again, I think it was 2018. We had a very prominent politician here. This is Mitt Romney. He actually ran for president against Barack Obama, I believe, in 2008, if I'm not mistaken. And so I teach a class in air quality engineering. And so I use this image of Mitt Romney because it was a part of a social media meme back in 2018, where his staff presented him, as you can see here, with a birthday cake made out of Twinkies. And instead of doing what most people would do, which is to simply blow out all of the candles en masse, he proceeded to pick up each Twinkie one by one and blow out each candle individually. And so that struck the online masses as being particularly strange and weird. And so he was roundly criticized for being strange and weird. But actually his response was that he was suffering from a cold and he didn't want to transmit his, he didn't want to shed virus or bacteria onto the Twinkies and then get other people sick. And so I took that, that social media storm and I turned it into a homework assignment. I had students develop a fluid dynamic model representing, someone blowing potentially infectious aerosols onto a circular cake here and looking at the distribution pattern, right? So I tend to have a lot of fun when I can with how the areas of our research get represented in the public sphere. But long story short, nowadays there's really very little need for me to preface. Thank you. 2012. Yes. So Mitt Romney ran for president against Barack Obama in 2012. So there's really no need to preface why we need to be concerned about airborne disease transmission because of COVID, right? And so really, the bigger question is, how did we get to a place where we're considering the intersection of plasmas and airborne viruses? And then more importantly, how did I get there, given my background in aerospace engineering? So I spent the first 10 to 15 years of my career still studying aerosols, but studying chemical aerosols produced as a result of combustion. And what you're looking at here are various perspectives of a device called an electrostatic precipitator. So these are devices, very large devices that power plants use, especially coal-fired power plants, to remove particles and ash from their combustion gases before those gases are released to the atmosphere. And the basic principle is you charge the particles prior to those particles, as they're suspended in a gas, entering an electric field, and then the particles drift in response to the electric field until they are collected on a solid surface on one of the charged electrodes. And so in that way, you have the ability of removing particles from a gas stream without use of a filter. And again, I think pre-2019, the importance or the distinction of filtration versus non-filtration would be a fine point that not many audiences would get. But I think, in the past 2.5 years, we have all become very familiar, perhaps overly familiar, with what it means to have an N95 or KN95 mask on your face and having to breathe through it. And so electrostatic precipitation is favored for these industrial processes precisely for the same reason that we don't like N95 and KN95 masks. That being that the removal of particles by filtration inherently reduces the throughput of air that can accompany that process. So the same way that we have difficulty breathing through masks, power plants have difficulty forcing their exhaust gases through filtration media, and so electrostatic precipitation is favored. And so upon arriving at Michigan in 2012, it was that background in coal combustion and electrostatic precipitation, when combined with my colleague here, Krista Wigginton. Her background is in environmental science and wastewater treatment. And so at the time, she was looking at virus persistence during wastewater treatment processes. I was looking at mercury emissions from coal-fired power plants and the role of electrostatic precipitators in reducing mercury emissions. And so really it just came down to an openness to collaborating with others and the opportunity to do so. So it really was just an informal meeting between Krista and I one day, not scheduled, not on the calendar, just a Friday afternoon, just sitting and chatting to young and new faculty, exchanging ideas about their research areas. And that's how we arrived at the intersection of our research areas. So the electric field and the cold plasma components of electrostatic precipitation taken from my research, and her virus survival, virus persistence taken from Krista's research, we came to be interested in, and again, this is 2012, so this is pre-pandemic, we came to be interested in what could we do to better understand ways in which non-thermoplasmas could be used to sterilize ventilation air and to prevent airborne transmission of infectious pathogens like viruses. And so just as a little bit of background, non-thermoplasmas, I'll shorthand them as NTPs in the rest of this presentation. They are a cousin of combustion flames. So the flame that you have on your natural gas stove is a thermoplasma. A non-thermoplasma is similar, it's just that it is not fueled by a hydrocarbon fuel and it is not hot. But it has many of the same chemical characteristics as a thermal flame, just without the high temperature. And so non-thermoplasmas or corona discharges produce radical species, highly reactive species that have the ability to attack bacterial cell membranes and virus capsids, leading to the inactivation of viruses and the ultimate death of viable bacteria. And so non-thermoplasmas have been used. And so you see pictures here, you see a non-thermoplasma being used to sterilize strawberries, sterilize fruit. Here you're seeing a non-thermoplasma being directed at a fingertip. There have been uses of non-thermoplasmas to treat skin disorders like eczema. And so, you know, for sure, non-thermoplasmas in medicine or non-thermoplasmas used for sterilization, this is not a new area, but I would say that these applications have a longer history than the use of non-thermoplasmas for sterilization of a flowing airstream, in part because, you know, the configuration is much easier here. If the device, if the surface needing to be sterilized is static or motionless, you know, it becomes much easier to do quantification. It becomes much easier to control the conditions and to know quantitatively the degree to which viable viruses or viable bacteria have been reduced by this exposure. And so for us, the challenge that we were facing was it becomes much harder to quantify the degree of inactivation of a virus or a bacterium suspended in an airflow when the virus or bacteria are aerosols that are flowing continuously past or continuously through the plasma region. And so there certainly is a history of plasmas used to sterilize static surfaces. Our contribution is, or has been, demonstration of plasma inactivation of bacterial and viral aerosols suspended in an airflow. And so just giving you a schematic representation of one of our experimental setups. You can see here we have a packed bed. This is a cylindrical tube, a section of which we have filled with dielectric beads, such as borosilicate glass. And then there are electrodes on either face of this packed bed, as well as a belt electrode encircling the packed bed. And so between these three electrodes, one electrode at a high voltage, the other two electrodes at ground, we generate a plasma that that penetrates through the packed bed and propagates through the interstitial spaces between these beads. And so through this packed bed non-thermoplasma reactor, we flow a continuous flow of air that has been seeded with viral aerosols. We generate a fine mist of a virus that, fortunately for us, is harmless to humans, but is lethal to E. coli bacteria. And so it's called bacteriophage MS2. And so it's easy to work with precisely because it is harmless to humans, but can be easily tracked or easily assayed by looking at its effect on a lawn of E. coli bacteria grown on a petri dish. And so we generate these aerosols from a buffer solution, saline solution. Those droplets evaporate in this airflow before reaching this plasma reactor, and we simply sample upstream of the reactor and downstream of the reactor into these twin impingers, samples of those aerosols so that we can capture before and after plasma treatment these viral aerosols, again, in liquid solution. And then we take samples of this liquid solution and then we plate them on lawns of E. coli bacteria in petri dishes. We incubate those for 24 to 36 hours. And then what results at the end are petri dishes in which what was once a continuous surface of E. coli bacteria now has pockets where the bacteria have died as a result of exposure to the bacteriophage MS2 that is infectious to the E. coli bacteria. And so what you'll see in our results are plaque assays, basically, so an assessment of the number of those pocs, the number of those open pockets on a given Petri dish that were counted manually as a result of exposure to different samples taken from these two impingers. So obviously, if we are perfectly effective in inactivating all of the airborne viruses coming through the plasma reactor, then taking a liquid sample from this impinger and depositing it on a lawn of E. coli would result in no development of no pocs, development of no open pockets in that lawn. And then the more viable virus you have, the more pocs or pockets develop in the lawn after incubation. And so you can see here a close-up of that plasma reactor. Again, it's cylindrical in shape, which is basically a cylindrical plexiglass tube, a section of which we have filled with borosilicate glass beads. And then those that segment of beads is faced on either end with ground electrodes and is encircled by a positive high voltage electrode. And so here's an image of it. The setup overall, you can see a power supplies, oscilloscopes, the induced draft fan to promote the flow of air through the plasma reactor, which you can see here. And then in the inset, you can see a close-up of the plasma reactor as it's energized. As you can see the characteristic indigo glow of the plasma at high voltage. So this is what a non-thermal or cold plasma looks like when it's energized. And so in the next slide, I'll basically show you three separate results at three different locations, each of which is representing either data taken from plaque assays, and that plaque assay is a measure of the concentration of infectious virus. And there's also companion data taken at the same three locations where we're not measuring infectious virus, we're simply measuring the abundance of the viral genome. Okay, so we have two different measures. We have one, we're saying, okay, how much, what percentage, how many log reduction are we achieving by exposing these viral aerosols to this non-thermal plasma in the reactor? And the second measurement, the measurement of the abundance of the viral genome, is simply saying, irrespective of whether the virus is infectious or not, how much of both infectious and non-infectious viruses do we have upstream and downstream? And so that second measure, the abundance of the viral genome, is intended to as a control so that we can capture the amount of virus that gets filtered out in this packed bed, right? So we don't want to count in our measure of virus inactivation, we don't want to count simply the process of the virus being filtered out as a result of impacting the glass beads, or as we talked about earlier in the presentation, as a result of electrostatic precipitation, the presence of a charge inducing viral aerosols to stick to these surfaces in this packed bed. And so here are the results that I was referring to. So these last two collections of bars are the immediate upstream and immediate downstream results. And then this first set of results is basically our baseline. These are the measures of infectious virus and abundance of viral genome with the plasma reactor on in the darkest bar and the plasma reactor off in the hashed bar. And so I'll start with the lightest bars first. So the lightest bars here, here, and here represent the abundance of the viral genome. And again, I'll remind you that this is not a determination of whether the virus is infectious or not. It's just the presence of the virus, whether infectious or not. And so you can see far upstream of the plasma reactor, we have, these are log concentrations. So, 10 to the 6th genome copies per liter of air. And then as you get nearer to the plasma reactor, there's some dilution that has occurred. And so you have a lower concentration of genome copies of the virus, but still quite high, about 5.5, 10 to the 5.5. But you can see that the two columns are very close to each other. They are essentially, they're maybe half a log different with the plasma reactor on versus the plasma reactor off, the plasma reactor energized and the plasma reactor not energized. And so, we have a reduction in the genome copies abundance as you approach the plasma reactor. And then we have a small reduction across the plasma reactor, about a third reduction, a third of a log reduction that we attribute to filtration. But overall, you can see that there's very little differentiation between the plasma reactor on and the plasma reactor off in the upstream impinger samples as compared to the downstream impinger samples. So we see that we interpret this to mean that the energization of the plasma reactor really has little or no effect on the abundance of the genome copies that we measure upstream and downstream of the plasma reactor. And so the filtration is really primarily due to impaction and not due to any electrical effects. The darker columns are plaque assays. And again, the darkest column is plasma reactor on, the hashed column is plasma reactor off. We see the same or even a more dramatic drop in the abundance of infectious virus as we go from far upstream to the near upstream impinger. But we see a bigger drop off now across the reactor, right? So this is the upstream impinger, this is the downstream impinger. We can see that with the plasma reactor on, we have, again, a marked drop off, a 2.5 to 3.5 log reduction in the abundance of infectious virus with the plasma reactor on. With the plasma reactor off, we see very little reduction in infectious virus. And in fact, that reduction that we see with the plasma reactor off is on the same order as the same reduction that we saw in the gene copies, right? So that was what we attributed to filtration. So it is this difference in infectious virus concentration with the plasma reactor on that we attribute to inactivation of this airborne pathogen, separate from simple filtration of that pathogen in this packed bed. And so this was actually only the second study of its kind to demonstrate inactivation of an airborne virus using non-thermoplasmas. And even in this result, we reported this as 2.5 to 3.5 log reduction in infectious virus as a result of plasma exposure. But even that is a conservative estimate because the data that we got in the downstream impinger actually showed that there were no infectious viruses in the downstream impinger after plasma treatment. And so what we're reporting here is not the result of an actual plaque assay after plasma treatment. What we're reporting here is our lower detection limit, which is we're saying that the abundance of infectious viruses after plasma treatment is lower than the limit with which we can detect using our plaque assay method, so that the degree of inactivation, the number of logs reduction in infectious virus, is 2.5 to 3.5 log at a minimum. It very well could be more than 3.5 log. It's just that we cannot measure, or we were not able to measure via plaque assay infectious virus concentrations below this lower detection limit. Okay. And so we definitely continued that same approach, looking at different applied voltages. So you can see the log reduction in infectious virus at 12, 16, and 20 kilovolts. And you can see that there's a continuous upward sweeping trend in terms of the degree of inactivation of this airborne pathogen as you apply more energy, as you increase the voltage to the plasma reactor. What we're also seeing here is two different airflow rates. So the question of how quickly does the plasma inactivate the virus? So at the 170 liter per minute airflow rate, the airborne viral pathogens are exposed to the plasma for about 0.3 to 0.4 seconds. So it is quite rapid inactivation of the virus that we're seeing here. And we're seeing, very little effect of roughly doubling the airflow rate. You might expect as you double the airflow rate, you might see a dramatic drop in the effectiveness of the plasma treatment. And we're seeing, a small drop at the very highest voltages, but very little drop at the lowest voltages as a result of doubling the airflow rate. And so, there are other features of plasma treatment that are of interest if we're thinking about actual applications of the plasma for sterilizing ventilation air, that is air intended for people to breathe. One would be the power required. So plasmas, as I said at the outset, have been used quite widely for sterilization of surfaces, and plasmas have been used quite widely to catalyze other chemical reactions. So plasmas for material synthesis, so the transformation of hydrogen into hydrocarbons or things like that, using plasmas as a catalyst. That has also been quite thoroughly studied for several decades even. The challenge has always been to using plasmas for chemical synthesis is that they tend to be power hungry, so that to achieve the desired efficacy of that chemical synthesis process, the energy required for a plasma-based process is much higher than, say, using a metal catalyst or simply providing additional heat to elevate the temperature of that process. So while plasmas have been studied for chemical synthesis, what we're showing here in this top graphic is 9 or 10 studies taken from the literature showing the efficacy of their chemical synthesis process. These are all the gray symbols. So on the y-axis, the gray symbols represent the efficacy of some chemical synthesis process that is being catalyzed by a plasma treatment as a function on the x-axis of specific energy. So specific energy is watts of plasma power divided by the gas flow rate of the reactants in that chemical synthesis process. So watts per volumetric flow rate translates into joules per liter, so energy per liter of reactant. And so all the gray symbols represent various studies that have been looking at plasmas for chemical synthesis, and you see that they are all clustered above about 100 to above 1,000 joules per liter in terms of specific energy. By comparison, the red symbols represent our data in squares, and then the red diamond is the one other study that preceded ours looking at plasmas for biological remediation or plasmas for air sterilization. And you can see that all of those biological applications tend to cluster or tend to be spread over a range of specific energy that is much lower than has been used in the past for chemical synthesis. So the argument here is that the evidence suggests at this point that while plasmas might historically have been viewed as energy intensive for chemical processes, it appears that plasmas used for biological processes, particularly airborne pathogens, are much more energy efficient, have 10 to 1,000 times lower energy intensity than for chemical processes, which is of course promising when you're thinking about the energy needed to sterilize ventilation airstreams. In the lower frame, what we're showing here is a production of ozone by plasma. So for sure, plasmas also historically have been used in air to produce ozone. So if the intent is to produce ozone, ozone itself is an oxidant and ozone can be a sterilizing agent, plasmas historically have been used to produce ozone. Now, producing ozone to sterilize a surface is fine, but before the space in which that surface, let's say that you have a room that you're trying to treat that has been, let's say a room in a home that's been flooded, and you're trying to treat the mold that has grown in that room. Certainly you can fumigate that room with ozone, but before you reoccupy that room, you have to remove the ozone because ozone is toxic to humans at high concentrations. And so another obstacle that has been present in the use of plasmas for sterilizing airstreams is a perception that the ozone generated as a result of that treatment would make that airstream unbreathable for humans and animals. And what this figure is intended to show is at the specific energy values characteristic of the processes that we've been exploring, the amount of ozone that is generated is much, much, much lower than the amount of ozone that typically would be generated in an an intentional ozone generator used to generate ozone, right? So we are not using ozone as our disinfectant in our plasma process. And for that reason, we have no desire, no interest in generating high concentrations of ozone in order to have a more efficient process. Our inactivation of airborne pathogens is occurring from direct exposure of those pathogens to the plasma and not from exposure of those pathogens to ozone that ultimately is formed downstream of the plasma. So, you know, we're definitely aware of and we're sensitive to the concern that ozone is not something that we would like to have as a residual in the air after plasma treatment. And so what this figure is intended to show is the effectiveness of ozone filters that you can find in many commercial devices. So your photocopier or your laser printer likely has an activated carbon ozone filter at the ventilation exhaust where the cooling air comes out from the device. So we know how to reduce ozone, how to remove ozone from airstreams. And what we're showing here is that a commercial grade ozone filter taken from a copier or laser printer, the blue bars represent ozone concentrations as a function of airflow rate through our plasma reactor before they pass through the ozone filter. The red bars represent ozone concentrations after the ozone filter, okay? And then the different bar shades, like the darker blue is a higher voltage, so you get a higher ozone concentration. The lighter blue is a lower voltage, so you get a lower ozone concentration. I'll draw your attention to the fact that the y-axis is actually a log scale. So this is more like the difference between 6 or 7 ppm and 0.5 or 0.6 ppm, the difference between these two bars. And so you can see we can get a very dramatic reduction in ozone just using a commercial ozone filter that you can buy for your laser printer and your photocopier. So removing ozone post-plasma treatment from an airstream is not a challenge. And what is shown by that dashed line is the regulatory limit set by the state of California for indoor air cleaners for their ozone emission. And so you can see without trying, in many cases, the implementation of a commercial grade ozone filter taken from a laser printer or a photocopier is able to reduce the ozone concentrations produced by the plasma to below that state of California regulatory limit. The state of California is the only state that has a limit on ionizing indoor air cleaners. There in the US, there is no federal limit for the amount of ozone that can be emitted by an indoor air cleaner. And so lastly, just to make a fine point or to put a fine point on the issue of ozone, this is a picture of one of those commercial grade ozone filters taken from a laser printer where we're viewing this fluorescent light fixture through this highly porous ozone filter. So the point here is that while yes, the plasma reactor still requires some sort of filter to remove ozone. So we can't categorically claim to be filterless in our air treatment process. The point here is that to the extent that you can see through the ozone filter, if you compare that to a HEPA filter, or if you compare that to your N95 or KN95 mask, the ozone filters are much more porous, have a much higher permeability, and therefore the obstruction of the airflow that comes with an N95 or KN95 mask material or a HEPA filter material, all of that flow obstruction is greatly reduced because the filter that we have to use is much more porous than the filters used for filtering out particles. So our objective is to inactivate airborne pathogens and then use without a filter, and then use a highly porous filter to remove the ozone, rather than taking the approach of N95, KN95 masks or HEPA filters, where the approach is to try and filter out these 300, 400, 500 nanometer sized particles, which requires a much more tight weave, a much less permeable filter media, and therefore much higher obstruction of the airflow and all the associated problems that we're familiar with filtration in general. So the response to that Journal of Physics D article was quite profound. We had lots and lots and lots of inquiries. The last 2 years has been quite hectic and chaotic for our group and for myself. We had outreach from a variety of sectors, anywhere from the MBTA, the subway in Boston. The researchers at Lincoln Labs reached out to us inquiring about the use of non-thermoplasmas as a way of providing protection against biowarfare attacks to public transport systems. And I imagine that there are, similar concerns in the UK with underground subway systems and their susceptibility or their vulnerability to biowarfare attacks. We had inquiries from physicians looking at using non-thermoplasmas to sterilize ventilators, right? So when the COVID pandemic was first starting, there was a shortage of ventilators, and there was an initial strategy of, instead of assigning one patient each to their own ventilator, because there was a shortage of ventilators, there was an initial strategy of using one ventilator to ventilate multiple patients and to avoid, and so there was an interest on the part of physicians at Stanford Medical Center. Could we avoid cross-contamination between patients that are sharing a ventilator by installing non-thermoplasmas to sterilize the airflows treated by the ventilator? And so there was a lot of, like I said, there from various sectors, a lot of interest in this approach to air sterilization that did not involve trying to filter out submicron sized particles. But our origins actually pre-pandemic were in animal agriculture. So before the pandemic, before humans saw a need to protect themselves from airborne pathogens, vigorously, really the most interest came from pig farmers and poultry farmers, right? So pig farmers and poultry farmers were quite aware and quite sensitive of the vulnerability of their herds to airborne pathogens harmful to livestock. And so this just gives a quick summary of the different livestock diseases that were of concern in animal agriculture. There's foot and mouth disease for cattle. In the US, the estimate is if we had a widespread foot and mouth disease outbreak, the impact on the economy would be a $60 billion cost. Avian influenza or bird flu back in 2015 in the US, we preemptively culled more than 50 million chickens in order to limit the outbreak to just a few Midwest states. More recently, it's a little less known, but of the species that are susceptible to SARS-CoV-2, so the current epidemic, are mink. And so while this is nowhere near as large a market as pigs and chickens, owners of mink farms have been quite impacted by the spread of SARS-CoV-2 from humans to mink as a zoonosis. And so for us, pre-pandemic, back in 2015, 2016, 2017, we were studying the effectiveness of non-thermoplasma for inactivating a particular airborne virus that inflicts pigs. And so this virus is called Porcine Reproductive and Respiratory Syndrome virus, or for short, it's called PRRS. And so in the US, PRRS costs pork producers about $650 million each year. It has more than a 95% mortality for piglets. It can cause spontaneous, it's actually very, very sad and very, very shocking. This is nothing, University of Michigan is not a school with is not a university with an agriculture school. That's actually Michigan State University, a different university down the road from us. And so I have no background in agriculture, but I learned all of these things in working with pig farmers as a result of their interest in finding ways to sterilize the ventilation air that they provide to their animals. And so in addition to the 95% plus mortality for young piglets, an outbreak of PRRS among pregnant sows, among pregnant female pigs, can lead to something that they called abortion storms. So the female pigs, the pregnant female pigs, spontaneously abort. And sadly, these abortions tend to come in waves within an animal housing structure. So you will have a barn full of pregnant sows, and over the course of several days, the vast majority of those sows have aborted their unborn fetuses. And so, highly lethal, highly impactful to the pork industry. You might say, as we've, kind of come, again, this is something that we, on the human side, 2016, 2017, 2018, we did not appreciate as much, but pig farmers and chicken farmers absolutely did appreciate the fact that this virus had a rapid rate of mutation. And so that variants would come out every year or two. And so you could vaccinate your herds of pigs, but over the course of 12 to 24 months, you would find that the vaccine effectiveness would wane, and you would have to have a booster shot, or you would have to get a new vaccine. So again, 3 or 4 years ago, this was an important factor in trying to find additional protective measures beyond vaccination for preventing the spread of this virus. And again, so 3 or 4 years ago, we had to make this point. In the post-pandemic world, it's abundantly clear that vaccines alone are not the road out of the current pandemic, because if you have a virus that is undergoing rapid mutation. And so, all of these things, high mortality, high impact, high death rate, of a virus that is either resistant to vaccines or that was rapidly mutating, so that vaccines were not a panacea. And then finally, again, 3 or 4 years ago, just as in the human case with airborne pathogens, 3 or 4 years ago, for pig farmers, they were certain, they had proven that the PRRS virus remained viable in the air. So there was not a question of, well, does the virus only get transmitted on surfaces? So for example, on clothing of farm workers, or on tools, or on feed given to animals. And so it had been established the PRRS virus could survive more than 4 kilometers distance from the initial release from an infected pig. So we had all of the components needed to look at something that would provide some protection for pigs and chickens from airborne pathogens brought into a pig structure, to an animal structure, along with ventilation air. And so pig farmers in the past had been using HEPA filters to filter their ventilation air, but they had been using them very grudgingly. And so if you look at the market of all US pig barns, so if we assume that a barn contains maybe 2,500 pigs, if we divide the total US population of pigs by 2,500, that gives us about 37,000 pig barns. Now, within that 37,000 pig barns, there certainly, there certainly is the thinking, or the logic is that not necessarily do all pigs need protection from this airborne pathogen. The pigs that are very close to being harvested, the pigs that will go to market very shortly, many of those adult pigs can survive a PRRS infection and recover and still go to market. And because PRRS does not survive or is not a threat in terms of pork as a food product, pigs that have survived a PRRS infection can still be harvested. So the pigs that are closer to harvesting would not likely be candidates for HEPA filtered barns. But you do have a subset of that. You have about 6,600 barns where you have high value pigs. So these are the pregnant sows that give birth, not only periodically, but give birth continuously almost. And you also have the high value boars. So you have the boars that provide the semen that are used to inseminate the sows. So those are considered high value animals. And all of those barns you would want to outfit with HEPA filters to protect those animals from airborne pathogens. And so within that 6,600 barn population, several years ago, 3 or 4 years ago, you could see that there was only about 120 barns that were using HEPA filters. So of the barns that could use HEPA filters, only a very small fraction were using them, in part because HEPA filters are hard to use, right? So all of the things that we complain about in terms of N95 masks, KN95 masks, are hard to breathe through. They become clogged, they become moisture soaked. All of those things apply to HEPA filters used to filter barn air. And so Nicola asks, do you have estimates for how much it would cost to implement non-thermoplasm in agricultural settings? Yes, we do. So actually this was the market case that we would have to make in terms of securing funding to do the research in an agricultural context or for agricultural applications. So there are two components that we typically presented. We typically presented the capital cost and the cost of ownership, right? So and so Nicola, this is actually an excellent question. So for non-thermoplasmas, because they did not introduce as much flow obstruction in the ventilation air, we projected that you could install non-thermoplasmas to treat all of the air sucked into a barn. And you could do that without having to retrofit the barn to be airtight. And so what do I mean by that? So hopefully you're familiar with the reasoning behind the general public in the current COVID pandemic. The reason why the general public has not been allowed or has not been assigned to wear N95 and KN95 masks. Those types of masks have been reserved for healthcare workers and frontline workers. The reason is that while the material of the mask is capable of filtering 300 nanometer size particles to 99.5% efficiency, that is only possible if the mask makes a perfect seal with your face. If there is an imperfect seal with the mask, then there will be leaks around the mask. And studies have shown that for someone wearing an N95, KN95 mask that has not been fit tested to perfectly fit their face, that the leaks of aerosols around the mask through those leaks reduces the mask effectiveness to being about the same level as just a loosely fitting surgical mask. So to the extent that the general public does not have access to fit testing in the way that doctors and nurses do, so doctors and nurses get assigned N95, KN95 masks in their hospitals, but then those masks are also purposely fit to their faces to make sure that they have the best possible fit. If you don't have the ability to fit those masks, then you don't have the ability to curtail leaks around the mask. And so the same type of logic applies to HEPA filters installed in pig barns. If you install a HEPA filter in the ventilation air ducts and you are drawing ventilation air into your barn by suction, right? So you're sucking air into the barn. If you install HEPA filters in a ventilation air system, you will restrict the airflow to that ventilation air system, and the air outside will simply find an alternate route to get inside the barn through cracks and crevices. And so installation of HEPA filters requires a relatively, to get to Nicola's point, requires a relatively expensive retrofitting of the entire barn to make it airtight. And so that retrofit is a high capital cost. And then HEPA filters also have high ongoing operational costs because the cost of the filters tends to be more expensive because, in fact, they are HEPA filters. And so for us, what we typically show in our commercial market analysis is that non-thermoplasm is used to sterilize ventilation air in agricultural settings, saves money both on capital costs because you don't have to retrofit the barn to make it airtight, and saves money on cost of ownership because the filter replacement for the ozone filter is about 1/10 of the cost of the filter replacement for HEPA filters. So great question, Nicola, and appreciate your asking that. And so, the argument, just to kind of summarize our the reasons the arguments we made for non-thermoplasma being a good replacement for a HEPA filter. If we start, so here we're comparing HEPA filter results taken from the literature to our non-thermoplasma results that we published. Starting at the bottom row of this table, we got about the same or greater degree of reduction in infectious pathogens. HEPA filters get about 90% reduction, 97% reduction. And again, part of that is the effect of imperfect seals around the HEPA filter and the ability of aerosols to leak around the filter if you have those imperfect seals. Whereas for non-thermoplasma, we demonstrated, as you saw, greater than 2 log reduction. But there are other benefits as well. The flow area, HEPA filters tend to be very large in cross-sectional area in order to treat a low velocity airflow. Our plasma module is about 70 times smaller in terms of its flow area, so it can be more compact. The pressure drop across our plasma module is about 25% lower than the pressure drop across the HEPA filter, while our plasma module is able to treat about 4.5 times the volume of air as a HEPA filter, right? So we're treating more air at a lower pressure drop in a much smaller compact footprint than is possible with a HEPA filter. And so in the interest of time, I'll move a little more quickly through the remaining slides. We have conducted tests. In fact, ours was the first ever demonstration of a non-thermoplasma inactivation of an airborne virus known to cause infection in humans or animals. So remember, the work that we had just been discussing was with bacteriophage MS2, which is infectious to bacteria, but is harmless to humans and animals. And it's for that reason that we work with it, because it's easy to work with. But in collaboration with the University of Minnesota, we took our same plasma reactor to their research institute, and we demonstrated essentially the same degree of inactivation of this PRRS virus that we were talking about, the same degree of inactivation at the same experimental conditions as we demonstrated previously with bacteriophage MS2. So that was the first ever evidence of non-thermoplasma inactivation of a virus known to cause human and animal disease. However, there are lots of challenges to working with actual viruses that cause human or animal disease. Viruses and bacteria, if you deal with biology, if you deal with aerobiology or microbiology, this is a whole different world than engineering. For sure, keeping the viruses alive is a challenge. So you have to use different formulations of a liquid that you then need to aerosolize into fine droplets, and then have those fine droplets evaporate so that you leave just the virus remaining in the airstream. It turns out that for the PRRS virus, that liquid formulation is different than for bacteriophage MS2. That liquid was more viscous and had a lower rate of evaporation, which led to that to the droplets of the containing the PRRS virus evaporating more slowly. And then those liquid droplets, instead of being evaporated by the time they reach our plasma reactor, you can see here from this image, the liquid droplets actually accumulated in our plasma reactor. So we had an accumulation of that liquid there. And so that confounded some of our experimental results, but we were able, once we got back into the lab, to demonstrate, basically to correct for that confounding. So basically we were able to demonstrate the degree of reduction in the efficacy of the plasma reactor as a function of the amount of liquid that we artificially add to the reactor, interfering with the operation of the plasma reactor. Just, like I said, I'll skip through these slides. Separate from the lab studies at University of Minnesota, we've done field studies where we've gone out to actual pig barns with a prototype reactor that we had built by a local prototyping company called Quantum Signal. Here's a picture of it here. It's designed to be rugged, designed to be taken out into the field. Here are pictures of us doing those measurements. So the objective of these field tests was to kind of get out into the field and demonstrate in a more natural environment the effectiveness of a plasma reactor for inactivating the PRRS virus. And so the way that we did this was we found a nearby pig farmer whose pigs had recently received the PRRS vaccine, but this PRRS vaccine in particular was not completely inactivated PRRS virus, but it was a modified live vaccine. So it's similar to what you might get, what we might get as humans for yellow fever, right? So if you get a yellow fever vaccine, it's one of those things that you have to keep it refrigerated, right? So it is not a completely attenuated virus. It is modified live virus, and for that reason, you have to keep it refrigerated. So our objective here was these pigs, having recently received the modified live vaccine, would go through, as we all do, again, we're very familiar now, a mild course of the PRRS disease after vaccination, and that during the course of that disease, they would be shedding the PRRS virus, and that as we set up our plasma reactor in the downwash of this air exhaust vent from the pig barn, we could siphon off a portion of that air, pass it through our plasma reactor, and then measure efficacy of the inactivation of the PRRS virus without some of those experimental artifacts that we experienced in the University of Minnesota tests. And so here, just another image, just showing kind of the degree of dedication that my grad students had to doing this type of work. Not sure if you're remembering back in 2018, 2019, there were several outbreaks of the polar vortex. And so these pictures were taken just at the start of the polar vortex here. So these are exceptionally cold outdoor temperatures that we're working in. Here, just showing an image of us wearing coveralls. And you might think that we're wearing these coveralls for biological protection, but in actuality, we're wearing these coveralls in order to keep the odor of pig manure off of our clothing. So that's one of the first things we learned from these field tests is when you go out there, wear clothes and wear shoes that you don't mind throwing away once you get back home, because the smell will permeate everything that it can get in contact with. I'll skip these next few slides in the interest of time. I'll get to what Francis talked about, kind of the entrepreneurial component of the work that we do. So we have a startup company, Taza Aya, that is commercializing the technology that we've developed using non-thermoplasmas. If you remember, we said that the non-thermoplasma reactor could be quite compact. It could fit into a 70 times smaller footprint. And so that makes it well suited for miniaturization. And so what the startup company is doing is we're miniaturizing that plasma reactor such that it can fit into a small module that you might wear around your head in a visor. And by having the plasma reactor sterilize a stream of air about your head, we have proposed, and we've submitted patents to this effect, or patent applications to this effect, that we could provide a replacement for N95, KN95 masks that does not restrict the airflow, that does not restrict your ability to eat or drink while you're protected from airborne pathogens, because instead of trying to filter the airborne pathogens from the air that you breathe, this visor would produce a focused curtain of air that would be directed downward in front of your face and would deflect airborne pathogens in the environment around you from reaching your breathing zone. So in this way, you could wear the visor, the jet of air would be sterilized by the non-thermoplasma modules, and you would not have the breathing restriction of trying to inhale through the impermeable filter material. And you could also, for example, eat and drink while still maintaining respiratory protective conditions. Other studies that we've been involved in, and again, I'll just skip through these. We've been involved in studies of Legionella, Legionnaire's disease. So Legionnaire's disease is a respiratory disease caused by bacteria that are left as a residual in drinking water. And so it can be aerosolized from showers or fountains or hot tubs or the sprays of misting water produced in supermarkets to keep produce fresh. And so we paired with a researcher in the med school trying to understand why it is that of the 6 or 7 serotypes of the Legionella bacteria, only 2 specific serotypes are responsible for the vast majority of Legionnaire's disease outbreaks. And so we built a aerosol chamber, and we aerosolized the 6 different serotypes of Legionella bacteria, and did show that there were 2 specific serotypes that survived the aerosolization process. It survived longer as an aerosol in a droplet than the other serotypes. So that begins to start to explain why, of the different serotypes that exist in bulk drinking water, only these 2 ultimately survive in air to cause Legionnaire's disease as a result of the bacteria being inhaled. Lastly, we, of course, both in terms of COVID and in terms of influenza, there's lots of interest in the rate at which viruses become inactivated in air. One of the things that is not as well known in the current pandemic is that it really is only quite recently that scientists and engineers have been able to sample viruses from air and detect their viability. So you remember in the first few slides, we took impinger samples to collect the bacteriophage MS2, and we could measure the abundance of the infectious MS2 phage in those samples. Well, it turns out that for other viruses like COVID, like influenza, those viruses are much harder to collect from the air and retain their viability, retain their ability to infect. So that when, the vast majority, almost all the studies that have been reported historically looking at influenza virus in air, almost all of those studies have really only been reporting the abundance of the influenza virus genome in air, right? So the genome could be infectious or it could be not infectious. And so there really only recently have there been instruments developed specifically with the capability of gently, if you will, collecting influenza virus from air and maintaining or preserving its infectivity such that we can now measure the abundance of infectious influenza virus in air, separate from the abundance of just the number of influenza virus genome copies in air. Okay, so when we talk about influenza in air or airborne viruses, it's only recently that we've been able to begin to measure the abundance of infectious virus in air and how airborne viruses can become inactivated naturally just a result of for example, desiccation or drying in air. And so this has been studied primarily in outdoor settings, looking at the effects of temperature on how long viruses survive on surfaces, for example, the effects of relative humidity, so desiccation, how long viruses can survive as they dry on surfaces, and the effect of UV light or sunlight, right? So, but one of the things that's been studied much less thoroughly, and actually the originators of this concept were researchers at Porcine in the UK, is this concept of open air factor, the fact that viruses in a lab context, viruses in laboratory air were found to maintain their infectivity over longer periods of time than viruses in outdoor air, in naturally occurring air, in polluted air. And so without a specific chemical compound that they could attribute it to, early researchers back in the late 60s and early 70s called this differentiation, the different survivability of viruses in synthetic indoor air as compared to natural outdoor air. They called that the open air factor, or OAF. And so, as I said, the very first researchers in the late 60s, early 70s discovered this phenomenon doing research at Porcine in the UK. And what you're seeing here is you're seeing different concentrations of outdoor air pollutants, with the higher the concentration of outdoor air pollutants leading to the more rapid loss of viability of different viruses over time. So the y-axis is percent of the viruses that remain viable over 20, 40, 60 minutes. Similarly here, loss of viability over time. That loss of viability is accelerated as you increase the percentage of mixture in air that is coming from open air as compared to coming from a synthetic laboratory source. And so the thinking had been that there's some component in outdoor air that is responsible for inactivating viruses that doesn't exist in indoor air. And so one of the things that we're researching now, and we're just about ready to announce a $10 million center to study influenza transmission in indoor environments, is understanding now, not so much in outdoor air, but studying how chemical compounds in indoor air might lead to an effect analogous in indoor environments to the open air factor in outdoor environments. So for sure, in indoor air, previous studies of UV sunlight are irrelevant. Sunlight does not exist to a large extent in indoors. Indoors, temperature and relative humidity are quite tightly controlled. So there is not a lot of ability to adjust temperature and relative humidity in indoor environments to affect the rate at which viruses released in indoor environments, the rate at which they lose their viability. And so for us, the very interesting thing is taking the open air factor, taking OAF indoors and starting to study the sources of indoor air pollutants that could contribute to an indoor version of open air factor, and understanding how different compositions of those indoor air pollutants can be optimized so that we accelerate as much as we can the rate at which viruses released in indoor environments are inactivated, so that we can limit the spread of viruses indoors. And so things like photocopiers and laser printers, as I mentioned before, these are sources of ozone in indoor air. Natural gas flames are sources of NOx. All of these are oxidants that are known to have virucidal and bactericidal properties for viruses and bacteria. And so for us, we're really excited to begin studying kind of the indoor version of open air factor that was discovered more than 50 years ago in the UK. And so with that, I apologize for going long. I very much am appreciative for all of your attention. I'd like to acknowledge all of my collaborators at University of Michigan, University of Minnesota, all of my grad students and postdocs, and the companies that have helped us with our commercialization. both at the University of Michigan and at the startup company Taza Aya. We are hiring both for postdocs at the university and for staff positions at the startup company. So if you know anyone who's interested in these types of studies, please send them my way. And with that, I would be happy to take any questions that you may have.
Thank you so much, Professor Clack, for that presentation. No worries about it going slightly over. I think it was really interesting and fascinating the whole way through. Some really phenomenal ideas, and I'm really excited for the future of the projects and the innovations that you're coming up with. One of the questions that I have, I'm just curious why it seems like your non-thermal plasmas are so much more superior to the HEPA filters, why they can't be more widely adopted, or what we can do to try and accelerate it. I'm just curious what some of the roadblocks are that you envision towards scaling in the years ahead to try and make that a bit more widely adopted.
Yeah, I mean, that's a great question. And I won't even pretend to be completely unbiased, right? So, you know, I do have a startup company, and the non-thermoplasma technology is what we're commercializing. So I have an opinion and I have a perspective. You know, I think historically, you know, filters, you could go back centuries, right? If you know, again, when we're thinking about the pandemic, you know, there are images in the public health literature of these, what is it? There's a play there, and I can't think of it if it's, there's a play where people are wearing masks, and the masks are elongated, looking like bird beaks. And they look like that because they're filled with kind of these aromatic herbs designed to mask the smell of decaying bodies during, you know, one of these outbreaks of a pandemic or something like that. And so yeah. I think just, you know, physical filtration just has a long history. And so, you know, in the US and worldwide, it is just the thing that has been able to do what we're trying to do. And to the extent that we haven't had, you know, worldwide outbreaks of pandemics where we've needed something that worked better or didn't have the obstruction, to the extent that certainly for sure, you know, each N95, KN95 mask only costs pennies, right? You know, what we're envisioning, you know, our device will not cost pennies. It will cost much more than that. So there is an argument that masks are probably the most democratic, the most, you know, cost-effective approach to respiratory protection. But in cases where people do want or do need the ability to access their face, if they want to be able to eat in restaurants or drink in bars while maintaining this protective bubble, then this is something that a face mask can't do. So that's my attempt at an unbiased explanation. The more biased explanation, I think there's an interesting dichotomy between the US and the UK. So in the US, there is an aversion to ozone that I think does not exist in the UK. So I have colleagues in the UK that have companies. There's Christopher, what is his last name? He's at Manchester, he's retired, Whitehead, Chris Whitehead. He's at Manchester. And so there's a company that started decades ago building ozone generation systems that would be installed in, for example, fast food restaurants as a way of controlling the buildup of grease in their ventilation system. So there appears to be kind of what do you want to call it? That the industry has been segregated into turfs. That in the UK and the EU, there's kind of more acceptance of ozone. And interestingly enough, there is not very wide acceptance of UV. So there's this, you know, when I talk to my colleagues, they tell them, yeah, we can't have UV devices anywhere because of the fear of exposure and radiation and things like that. And in the US, it's the exact opposite. There's, you know, HEPA filter is widely accepted, UV is widely accepted, but ozone is seen as being risky and problematic. So my sense is that there's, you know, somehow among the industries, they've divided up the Western world where, you know, on one side, you know, one industry will be, you know, looked at and disparaged to the benefit of the others. And on the other side, the other industries will be looked at and disparaged to the benefit of the others.
Okay, so yeah, there's reunification of the different the industry as a whole to try and progress it further as well.
Sure.
Yeah, thank you. Yeah, that's really elaborate and gives real insight. Are there any other questions from the audience at all?
Adolphus has his hand up.
Adolphus, please unmute yourself and go ahead.
Hi, good afternoon. Thank you so much, Francis. And thank you so much, Professor Clark, for your presentation. I really enjoyed the discussion today and the contents that you're presenting. I really enjoyed them all. And I mainly have one question regarding the patented the jet curtain, air jet curtain design that you talked about. And I just find it was a very interesting concept and idea. And, my main question is mainly like, what is the projected cost of this product if it's made available commercially? And what, I think I might miss this part, but what is the largest particulate matter size that this curtain jet, air curtain jet could actually like sort of like, like filter out from our like inhalation?
Sure. So, yeah. As a startup company, the costs are going to be driven in part by, well, the minimum cost is going to be driven by the cost of materials, right? So fortunately for us, we have no rare earth elements. We have no expensive catalysts like platinum or things that you will
nice.
also. I'm sorry?
Sorry, that's a nice.
Yes. So, there's not, there really aren't really any expensive components to the device. So that sets the minimum price. Again, this is where we're really not, one of the many ways we're not competing with N95 masks. We're not setting the price at pennies, okay? And so the maximum price would be, what the market will bear, at least initially, especially for those who, really either can't wear a mask, right? Because they have breathing issues, or they just don't want to wear a mask. In the US, we've just ideated on an additional market, so for people in the US, I don't know to what extent you have this in the UK, but there's a political segment in the US that argues that face masks are an infringement on free speech. And so this could appeal to that market and allow them to have respiratory protection while still protecting what they see as their free speech, right? Right. And so the maximum price certainly would be tens, if not over $100 per unit. So yeah, it is not intended as a mass market, something that could protect the world. And so in that way, we're not competing with N95 or KN95 masks. To your second point about what is the largest particulate that the mask can protect against, it really is dependent on not only the size of the particle, but also the density of it and its inertia, right? So the other way in which we're not competing against N95 and KN95 masks is the fact that this device is designed to be for a layperson. It's designed to be for the masses. It is not designed for use in medical contexts, right? So if you are a physician, if you are a nurse, if you are a doctor working in a high risk environment where you have a substantial risk of exposure to COVID and COVID in high concentrations, then yes, N95, KN95 will still be the gold standard. But it is in those other contexts outside of the medical context where our device would be preferred. And so the question is, okay, what exactly is the risk at a restaurant, in a bar, on the street? And so what we're designing the mask to do is to protect against airborne particles, airborne pathogens in the ambient air, right? So for sure, if someone walks up to you and sneezes in your face, the amount of air that would be needed to deflect that impulsive jet is not something that we would want to operate at continuously, right? Conceivably, we could have different settings where you accelerate the velocity of the airflow. For example, if you're running in the outdoors rather than walking, or if there's wind or something like that. But if someone sneezes in your face, this is going to provide limited protection. We had people, when we presented this idea, we've also had people ask, well, does it protect against you touching your face? It's like, okay, yes, no. If you touch someone else, if you shake someone else's hand, who's just sneezed into their hand, and then you touch your eye, yes, our device will not protect against that. So, this is a limited degree of protection, but not, and so in that way, we're not intending to compete against N95 or KN95 masks. But to your point, they're really, outside of a sneeze or a cough, the inertia of airborne viruses in ambient air is actually really quite low. It's just like lint in the air. So there is not a very large, we've looked at fractions of a meter per second in terms of the velocity of that air curtain that's needed to deflect. And it's quite effective in deflecting airborne pathogens that are floating in the ambient air. So that is the primary kind of use category. If we're talking about things where the velocity of the wind is higher, or you're trying to protect against someone who's sneezing in your face, then yes, you would have to have a higher velocity air curtain in order to protect against those jets.
Right. Yeah, thank you so much for your clarifications. And yeah, once again, great presentation. I really enjoyed everything.
Thank you.
Have a nice day.
Yeah, just to make a comment on that as well, I was under the impression that these masks didn't necessarily protect you, but protected you from spreading to other people, right? So having something like your innovation might cost more for people to purchase, but if it gives you more protection, I would assume that the people that are more at risk or vulnerable or at concern of catching something like COVID would pay a higher premium for that anyway, would be my impression.
Potentially, yeah. So, I mean, for us, the interesting thing, and these are things that I've only learned as a part of kind of grow the startup company, is, you asked at the outset, why is it that we, and this is an argument that we make all the time when we're pitching our technology. We've spent in the US, we spent tens of billions of dollars on vaccines. And yet the leading advice from our CDC in terms of respiratory protection is, especially respiratory protection indoors, is to open all the windows in your room, right? I mean, it seems like there's there's an imbalance there in terms of where the investment has gone. Far more investment on pharmaceuticals and vaccines than on these types of things, right? But for sure, like I said, for N95, KN95 masks, if they are fit to your face, that is the gold standard of protection. And if you need that, and if you're willing to persist with the discomfort that comes with that, then yes, that is the gold standard. And that will protect not only you from the people around you, but it will protect the people around you from you. In terms of like a surgical mask, then it's less effective, right? So it's less effective in protecting you from the people around you, and it's certainly less protective in protecting the people around you from you. And you can see that really around the cloth face masks as well, that when you exhale, it is not so much that your exhaled particles get caught in the cloth mask, it's just that, or in the surgical mask, it's just that they get diffused, they get jetted in different directions in such a way that it reduces the exposure of people around you to your exhaled breath. And so to the extent that any of these masks have been certified by the US FDA based on particle filtration, one of the things that we're aware of in our company is that our approach, being something different, will require what most likely would be a longer, more involved FDA certification process, because there's nothing like it already, right? So that's the other reason why, the reason we have many different types of filtration products and many types of masks based on filtration is because it is easier and faster to get FDA certification on a technology where the FDA already knows what it does and already has protocols set up to test it.
Yeah. Spreading your eggs into different baskets so you don't kind of potentially go all in on one product that the FDA doesn't approve.
Right.
Yeah.
And so it's not that the FDA would say, we don't believe you. If they will say, okay, you prove it. You develop the technology, you develop the testing protocol, and show us that your device is as effective either as you claim it is or as effective as the incumbent products of N95K and N95 mask. Yeah. So yeah, there's that's the trade-off. It's like, yes, innovation is great and it's fun and it's really, it's exciting to create new things, but the trade-off is getting those ideas into market. New things need to be proven as being efficacious.
Yeah.
And you don't get to ride the coattails of everyone who came before you who developed something nominally similar to what you're doing.
Yeah, absolutely. Yeah, so the risk is high, which I guess is a good segue into perhaps the question that you wanted to ask that your colleague suggested.
Yeah, so I have a colleague who is also similar to you, Francis, a background in aerospace engineering, but is now doing kind of risk analysis. And without revealing his identity, COVID has been, for a lot of people, myself included, COVID has offered lots of opportunities to paraphrase, the most recent Nobel laureates, to do natural experiments, right? So, this is what the Nobel Prize a few days ago in economics just was awarded for 3 researchers, that laid the foundation for doing natural experiments. And so I have a colleague of mine who, has done risk analysis of COVID infection in classrooms here at the University of Michigan, looking at, okay, if all the windows are open, or if we have one air purifier set in a corner, or if we have the HVAC system turned up to its maximum value. And what's been interesting and eye-opening for me and alarming for him is that, for him in doing a risk analysis, he has been reporting and he has sought to publish all the possible outcomes, right? So risk of infection, risk of severe disease, risk of hospitalization, and risk of death. He has actually been barred by the university from publishing anything that includes that last category. And so I'm wondering, is there anyone, on your end, have you had similar types of pushback or similar types of concerns from institutions when considering kind of, okay, the efficaciousness of different types of indoor air or different types of transmission reducing approaches? Have people been reluctant to, if that last category is not 0, which it usually isn't, assuming these technologies are not 100% effective. If that last category isn't a 0, are you still okay to publish, or have you had any well, say this, but not that? I'm curious.
Yeah, that's a really interesting question. I'm probably not very best suited to answer that, seeing as I'm dealing with indoor plants and growing kind of crops and herbs and such. But maybe Nicola you might have had some experience with that, or I could also pass you on to some of my colleagues that aren't here today that maybe are dealing with more risky things like dam tail failures or nuclear power, where there have been issues with the risk of death actually being a lot higher than other industries. Nicola,
I actually have a little bit of experience with that, but I mean, it's on the record, so I wouldn't want to be very specific. But I think as soon as you deal with something that is not only public health policy, but general, some kind of public policy, so you will see pushbacks because it's just in its nature is political. So it relates to how people make decisions, and then you it's not only that you don't need to deal directly with politicians, but you just work with these people who are in services, and they kind of know how it works internally, and they know that media can take something that you said in the paper, and then they already think about like, oh, okay, so if you say this in the paper, then someone can write an article that will say this and that and blah blah blah. So and make it much bigger deal, you know. So I guess you always have to be cautious whenever you kind of talk about things that, you know, can be turned around and can be sort of interesting for media, I guess.
Sure. I mean, I think that's exactly the, you know, the conundrum that my colleague finds himself in. I think for us, it's just been a little bit disappointing to not have that pushback come from an industry. Well, you could say, so we're used to having that pushback from for-profit industries, right? It just so happens that in this case, the industry is our university that we work for, that we had thought, you know, would prioritize freedom of thought and freedom of expression over protecting kind of, or you know, trying to avoid these types of pitfalls of working with the media. So I think that's, for us, it's just been kind of disappointing to see that.
Yeah, it is a bit shocking. And I mean, at the end of the day, science is about trying to seek truth, right? And if you try and review your data and it's not something that you want to hear necessarily, that shouldn't avoid you from having to publish that.
Right.
Yeah, I don't know whether anyone else has any thoughts to chip in on that matter. Vladimir, Conor, perhaps not.
But yeah, I don't know. I think there have been a lot of controversies. I mean, we had all of the universities were making sure they were saying, "Oh, it's safe," getting all of the students to pay for their accommodation a year in front, and then, you know, a few weeks later, suddenly it's not safe, and all the teaching goes back online. So I think the academic sector's not come out of COVID with much glory at all, has it? In many ways. But then on the other hand, I mean, if you look at the scientists and the, you know, the vaccination people, they've done great things. So yeah, it's a difficult one, isn't it?
Yes, absolutely.
Yeah, it really depends on the university, I imagine. But I'll definitely forward your talk on to some of my colleagues that also are working in epidemiology and see whether they've encountered any similar issues. I know I had lots of bottlenecks, even getting ethical approval to interview people about their businesses throughout my PhD.
Sure,
so just in terms of getting publishing data that isn't something that they want to have their name associated with as a risk where it might get misconstrued or something like that, I'm not really sure what the best way to work around that is, apart from my approach is it's always better to seek forgiveness than permission. But you know,
spoken like a trailblazer, right? never look backward.
Right. So just wrapping up, I'm not sure whether anyone has any last questions before we go. And if not, I would like to give everyone the opportunity again to check out Professor Herek Clack's website and check out his positions that are available in his company, if you know anyone that might be interested. And just give our speaker a round of applause for an amazing talk, and thank you so much for your time.
Thank you so much. It's really been my pleasure.
All right, perfect. Well, I'll catch up with you after the talk, and I'll talk to you soon. Everyone have a lovely evening. Right. And great afternoon to you.
Thank you. Have a great afternoon.
All right. Thank you very much.
Bye.
Thank you.
Thank you.